I have to compliment you on how nice and kind your border control people are. Because, as you heard, I come from Malaysia. And when I had my first passport, the Malaysian passport said that you couldn't visit Russia, all the communist countries, and so on and so forth. And then more recently, they changed it to you can't visit North Korea and Israel. The last time I had a passport renewed, there's only Israel left. We are supposed to be a moderate Muslim country, but on some things, they're not that moderate. So anyway, your customs, your border control people were really nice. They asked me all kinds of questions, how many children I have, how much money did I earn as a doctor. But finally, I think what really got me into Israel was they said, "You're a doctor?" I said, "Yes." "You're a neurologist?" I said, "Yes." And they said, "Oh, good, okay. You can come into Israel." Maybe if I said as a cardiologist, they might not have allowed me in. But I'm pleased to be here, and I'm also very pleased to be able to share with you the clinical evidence for the efficacy and safety of NeuroAid. So the first thing to say is that all of us know that our patients after a stroke can recover in different patterns. Clearly, if you had a very mild stroke, you would generally improve. If you had a severe stroke, unfortunately, most of our patients don't improve. It's these people here who have a wide dynamic range of improvement that we're trying very hard to maximize their recovery. And the real issue is how do we do that? Now, of course, there have been some exciting developments of late. The EXCITE trial showed that in chronic stroke, if you had patients who were randomized to constraint therapy, they would improve and the improvement would persist. But I think many of us know that constraint therapy is a bit difficult to accomplish. There's also been interest in medications for stroke recovery. There is evidence that selective serotonergic reuptake inhibitors can improve motor recovery after stroke. There's not just animal evidence, but also human evidence. And many of you are familiar with the recent FLAME trial, where in a small number of patients enrolled 5 to 10 days after stroke, there seemed to be a benefit for patients who were treated with fluoxetine. But I think it's important to remember that it's actually very small numbers, and this result needs to be confirmed, and confirmatory trials are ongoing. Nevertheless, it's encouraging that so much advance has been made into the pathophysiology of post-stroke recovery. We know that there is a scientific basis for post-stroke recovery, and therefore there are targets for us to work on. And I think one of the most important targets, of course, is neuroplasticity, the ability of our brain to reconnect and to reorganize. And of course, what we want to know is how we can enhance this neuroplasticity in our patients. We also know that not only is there a spatial difference between the molecular and cellular changes in stroke, of course, we're all familiar with penumbra, core damage, etc., but we're also now increasingly aware that there is a temporal course to how we recover after a stroke. There are different mechanisms, and these mechanisms kick off at different times after a stroke. You can see that the growth-promoting genes all sort of come up and go down at different times after stroke. Dendrite branching peaks different times after stroke. So do cortical synapses and synaptogenesis. So what this means is that, as Professor Leginsky said, in our search for the magic bullet, for the magic target, we now realize that there are many targets, and that the targets are spread over a period of time. So not only do we need treatments which are timely, we also probably need multimodal treatments, because post-stroke recovery is a complex business. It is extremely unlikely that we'll find a single magic target. There have been several potential human restorative therapies that have been proposed: growth factors, small molecules, stem cells, more intensive physiotherapy and other cognitive training processes. There's a lot of interest now in robotics, and you may be aware that there's exotic treatments such as laser stimulation, etc. But what we're going to talk about today, of course, is whether there's a role for traditional Chinese medicine. And we know that we do need better treatments because we have a limited armamentarium for stroke neurologists and stroke physicians. We don't have any proven pharmacotherapy for improving stroke recovery. Why traditional Chinese medicine? Not only the work of Professor Lesinski, but many other workers have found that herbal supplements have many interesting pharmacological properties. They're antioxidants, anti-inflammatories, and also anti-glutamate effects. They can dilate blood vessels, they can suppress platelet aggregation, and they can improve, protect against ischemic injury. Moreover, if we could prove that herbal supplements are effective, it would have a huge impact on our management of patients because these supplements are already available and they have a reasonable safety record. What is the role of traditional Chinese medicine in China? Well, there are many traditional Chinese medicines which are currently in use with the approval of the Chinese regulatory authorities for stroke, so much so that it's widely accepted as a standard treatment for ischemic stroke in China. Outside China, the situation, of course, is very different. There's very limited acceptance because most of us are unfamiliar with traditional Chinese medicine treatment strategy. And I have to say, even after having worked in this field now for several years, I have not the slightest idea what my traditional Chinese medicine colleagues are talking about when they describe how they come to diagnose and treat patients. So I take a very pragmatic role. They obviously have discovered something. They can't describe it to me in Western scientific terms, but we can test it. There's also other technical issues. The inability to specify active ingredients makes traditional pharmacologists kind of uneasy. The variation in the formulation and concentration of products because of the processes also makes people unhappy. unhappy. And of course, more importantly in this day of evidence-based medicine, the lack of a reliable evidence base makes all of us uncomfortable. There have been several meta-analyses of traditional Chinese medicine. This is one from our colleagues, the Cochrane Center in Chengdu, where they analyzed many of the traditional Chinese medicines which are used. They found that there were a number of clinical trials reported, but the fact of the matter is that most of the clinical trials were really of very poor quality. Out of the 120 definite or possible randomized controlled trials, only 3 were randomized, double-blind, and placebo-controlled. Only 2 had long-term outcome measurements. And so the conclusion from these meta-analysis was that the trials were so poor that it was difficult to go and make a conclusion. Furthermore, the outcome measures were also difficult to go and, to understand. Only 2 trials assessed death and dependency. Most of the trials didn't report any serious adverse events or adverse events. Only 10 trials reported any deaths, and most of us who work with stroke patients can't believe that they were actually treating stroke patients. How is it possible that your patients don't die or have serious events? And there was a strong tendency to all report significant improvements, suggesting that there may be some bias in the reporting. A second meta-analysis came to roughly the same conclusion. All the problems with the trials, they were so unhappy with the quality of the trials that they said they were simply unable to perform a meta-analysis. So, not surprisingly, most people didn't accept that there was a reliable evidence base. So rather than just sit back and say, well, that's it, we decided that it was time to go and sort of re-examine the evidence base, especially for Neuro8. So as you've heard, Neuro8 is 9 herbal components, and there's some animal components which act mainly as an excipient. For this particular compound, there were some preclinical studies done before Professor Lesinski's work on different animal models of stroke, suggesting some effectiveness. there were long-term toxicity and short-term toxicity performed in animals, and there was also some clinical studies. As you heard, the pharmacology of Neuro8 is very promising. I won't go any further. And so what we decided to go and sort of do was to perform a meta-analysis of the treatment trials that were available for Neuro8 in chronic stroke. We used the standard Cochrane methodology to identify all the papers and found that we could identify 6 primary studies. 2 of these studies were reported in a pool analysis we published in Stroke, and these are the other trials which were done in Singapore as well as in Iran. All these studies were randomized controlled trials. The inclusion criteria varied in these studies in terms of age and also the time after stroke, but you will agree that most of these are in the subacute or chronic phase, 1 week to 6 months from stroke onset. The exclusion criteria varied, and the outcomes measures also varied, I guess, depending on whether you were a rehab specialist or you were a neurologist. I don't know if any neurologists use the Fugl-Meyer score. It's a very complex, comprehensive score, and I think that most of our rehab specialists would find the stroke scales that neurologists use kind of laughable in their simplicity. Our colleagues also used some surrogate endpoints, visual field deficits, as well as blood flow velocity. We did a risk of bias appraisal for these studies and found that the vast majority of these studies didn't have any methodological problems. And in the meta-analysis, we could show that in terms of functional independence, there was a benefit for Neuro8 in chronic treatment. And in terms of motor recovery, there was a near to significant trend towards improvement for Neuro8 as well. So all in all, this was very promising. Safety, there wasn't an issue. There wasn't an increase in adverse events for the treatment group, albeit there were very few outcomes. So to summarize, for chronic stroke, placebo-controlled studies had been done, but they were relatively small. The methodology is incompletely reported, but I think it's sufficiently robust. The clinical heterogeneity makes it difficult to go and come to a precise conclusion, but it allows us some generalization and generalizability. And the outcomes assessed were inconsistent, but they all pointed towards the same direction. So these were encouraging results in chronic stroke. And I think that the evidence base is reasonable for chronic stroke. Clearly, we do need larger trials, but the meta-analysis to date is positive. So why look at acute stroke? Well, one of the reasons is that the Chinese studies which looked at relatively acute stroke patients were not placebo-controlled. And the outcome measures that were used in China differed from those we know routinely used in international stroke trials. And most of the patients were recruited at a relatively late stage. So a group of us in Southeast Asia decided to go and sort of look at the possibility of doing a trial of Neuro8 in acute stroke. Because we believe that perhaps the first 3 months after stroke may be the most significant window of opportunity to enhance recovery of function. And that the pharmacology that you've heard about suggested that Neuro8 was a good candidate to be tested in acute ischemic stroke. CHIMES was a randomized double-blind placebo-controlled trial. You could get in the trial if you were less than 72 hours after stroke onset, and with the intermediate severity NIH stroke score of 6 to 14. 6 countries took part over Southeast Asia. The sample size was 1,100 patients, and the study treatment duration was 3 months. This map is curtailed. I think I've said this before. North of this line, everybody believes in Neuro8, so it's not possible to do placebo-controlled trials. I guess the Israeli version is that north of this line is Jerusalem, where everybody believes in something. may not be the same thing. I'm just struck at how diverse belief is in Jerusalem. And west of this line, Western countries, nobody believed in traditional Chinese medicine, especially the regulators, so we weren't able to go and start the trial. I don't know if there's an equivalent in Israel for that. Is there any atheist place in Israel? Maybe it's not allowed. Never mind, I won't go into that. So the study design, as with all academic trials, it's progressed slowly. The study design was proposed in 2005 when I was much younger. We managed to go and get funding from the National Medical Research Council of Singapore, and we initiated the trial very soon after that in Singapore, followed by the Philippines and other countries across Southeast Asia. And we reached the target of 1,100 in May 2012. I have to say that we were helped very much by an academic research organization in Singapore, Singapore Clinical Research Institute, which did all the monitoring and the statistics completely independent of the trialists. This is a group of all the hospitals that took part in the study across Southeast Asia. The majority of patients were recruited from the Philippines and Singapore. The Philippines contributed the most, but we had significant contributions from Thailand as well as other countries which took part in the trial. Lab safety was conducted in a subgroup of patients. There were no issues with laboratory tests or with ECGs, and this is published. Safety was very carefully looked at by our Data Safety Monitoring Board, which had 2 meetings and encouraged us to complete the trial to its target number. As you can see, we randomized 1,100 patients. 1 patient withdrew consent before a randomization, and we can't use the data, so you can see that we had roughly matched groups. In terms of patients not receiving treatment, again, they were matched, and so we were able to include large numbers in the safety analysis. We lost several people due to withdrawal of consent, etc., but again, it's matched between the 2 sites, the 2 groups of treatment. And we managed to go and sort of pull some people back with imputed results from last observation carried forward. So the intention to treat analysis encompasses more than 90% of the patients. In terms of safety, of the 1,087 patients who received study treatment, adverse events were similar between the 2 groups. Serious adverse events were also similar between the 2 groups, and there was no excess of deaths between treatment and placebo group. So the safety profile is really excellent. You can't calculate the number needed to harm because it's infinite. In terms of deaths, you can see it's equal. Serious adverse events is numerically not the same. And of the 10 most common SAEs, there is no excess in the treatment group. What's the primary outcome? So this was announced at the ESC this year. And as you can see, there's a tantalizing shift towards improved outcomes for the patients treated with NeuroAid, but this was not statistically significant. The point estimate is 1.09, but as you can see here, the confidence intervals cross 1. I think it's important to emphasize that this is a neutral trial. It's not a negative trial. We did not have a point estimate which is less than 1. In terms of secondary outcomes, the achieving MRS of 0 to 1 dichotomy, again, the point estimate is positive, but it's not statistically significant. And none of the other pre-specified secondary outcomes was statistically significant, although there was always a positive trend. Now, so you all know about this, the donuts. Are there donuts in Israel? It's kosher. Okay, good. So you can look at a donut, and some people will see the donut, and some people will see the hole. So to me, CHIMES is an important trial because this is the first time we've actually done a well-conducted, randomized, placebo-controlled, well-monitored trial to give us reliable results. So we should look at the subgroup analysis to see what we can glean out of it, because this is the first time we can actually look to see which subgroups could possibly benefit from this treatment. Prior to this, we didn't know. And you can see here that there are some subgroups which seem to improve or do better. Females, those who were treated, randomized later, more than 48 hours, and those who had a more severe NIH stroke score. And this I've said before, and this is important to emphasize because it gives us confidence to look at the subgroup analysis and generate hypotheses. And I think you also have to put CHIMES in context with all the other recent neuroprotective trials. There have been some neuroprotective trials which have actually turned out to be actually bad. So this SAINT-2 had a negative point estimate. And there are other neuroprotective trials which are truly neutral. So this is absolutely flat, and so is so ICTUS and CASTA are absolutely flat. Whereas CHIMES, there is a trend towards being positive. What about subgroup analysis? Well, I said that there was a interesting difference between those patients who were treated greater than 48 hours and those who were treated less than 48 hours. This is the group which was treated more than 48 hours but less than 72 hours. As you can see here, the treatment group shows a more positive trend. When you look at it statistically, you can see that by MRS shift analysis, the point estimate increases to 1.29 and is close to significance, as is the MRS 0 to 1 dichotomy. We took the group which was randomized after 48 hours and put it back into our meta-analysis, which I showed earlier of NeuroAid in chronic stroke, because we felt that patients who were randomized later could be classified as chronic stroke. And you can see here that at the end of the day, when you put it in, the result for chronic stroke is still positive. So I think CHIMES does not change the earlier conclusion from the chronic stroke meta-analysis that NeuroAid may be useful in patients with chronic stroke. Sorry, let's go back. And so many people ask, well, why could NeuroAid be, have a positive result in patients who were recruited later? I don't think it's because NeuroAid is toxic in the acute stage. It's because when we look at the patients who were randomized later or earlier, what we found is that the patients who were randomized earlier were not stable. In the trial protocol, it only asked for stable stroke patients, but it turned out that the investigators, including me, were not that good at predicting instability. Patients who were randomized earlier had more AEs, they had more infections, etc., etc. So I think that these patients shouldn't have been in the trial, and the effects were diluted. We also had the opportunity of examining, in a post-hoc analysis, early recurrent vascular events. And these include recurrent strokes, acute coronary syndromes, and vascular deaths. This is important because up to 17% of our stroke patients develop another vascular event or die from a vascular event in the first 3 months after stroke. Currently, we know that antiplatelets may be able to have an effect on preventing such untoward outcomes. And more recently, the CHANCE trial suggested that clopidogrel added on to aspirin may have an additive effect. We think that statins, angiotensin receptor blockers, and heparinoids may have an effect, but this is yet to be proven. So in the CHIMES trial, I think it's important to note that there was no difference in terms of concomitant treatment between the NeuroAid group and the placebo group. The vast majority of patients were on antiplatelet medications, were on statins, were on antihypertensive treatments, anti-diabetic treatments, etc. So these patients were all appropriately treated. There wasn't an imbalance. Nevertheless, when we look at early recurrent vascular events, the combined outcome, a composite outcome, showed that there was a 50% difference between the treatment group and the placebo group, 2.9% versus 5.6%, which was statistically significant. And when you look at the individual components of this composite, they all have a trend towards being positive for the neuro-8 group, reaching significance in fatal stroke. So I think this is an interesting result, which was unexpected from what we thought we knew about the pharmacology of neuro-8. But as Professor Lazinski has just pointed out, neuro-8 has many functions, including a function on preconditioning. So this may be one of the explanations for why neuro-8 could prevent early recurrent vascular events, although I think there may be other mechanisms involved as well. So as you can see, this difference in the groups, when you plot it out as a survival curve, is apparent very early on and remains constant throughout the trial. And this has recently been accepted for publication in Stroke. What about the subgroup analysis looking at stroke severity? Again, when you only look at the patients who were more severe, 9 to 14, again, there seems to be a trend towards benefit. And when you analyze it statistically by MRS shift analysis, the odds, the point estimate is 1.2, not significant, and the dichotomy is 1.45, again, not significant. But suggesting that perhaps more severe stroke patients would benefit from treatment. Well, why is this so? I take you back to the first slide that I put up. I think it's because if you have very mild stroke, you're going to get better anyway. And therefore, treatment with neuro-8 doesn't improve your chances of getting better. It's this more severe patients where a treatment effect may become more visible in a trial. But of course, the problem is that predicting the future is very difficult. I give you an example from Singapore because this is something that struck us periodically. We have very friendly neighbors in Indonesia who regularly during the dry season burn off all the vegetation. So this is not a typhoon. This is a smoke trail. Huge smoke trail which, driven by winds, passes over to Malaysia and to Singapore. You can't see Singapore, it's hidden by smoke. This is what the view from my office on a clear day, not a bad view. And this is the view when the smoke passes over us. Incredibly, our meteorologists were not able to predict from day to day, on hour to hour, what the haze was going to be like because they couldn't predict the wind. But I'll give you an example of how in chimes we can analyze to see that severity may have effect. So this is chimes just in the Philippines. Analysis was done on the request of our Filipino colleagues because they randomized the most patients, 46% of the total recruitment, led by Professor Joey Navarro. And this is the result from the Philippines. So remind you, this is the overall result, shift analysis, point estimate 1.09. In the Philippines, the overall result was a point estimate 1.38, which was very close to significance. Now, is this because there's something special about the Philippines? Perhaps. One of my colleagues there was convinced that the trial was going to be positive because every week she had prayed to Our Lady of Fatima and had given presents of eggs to the convent, and so she was sure it was going to succeed. Was it faith? Yes? Okay. Well, I think there are other more prosaic explanations. So just to look at the statistic again, for chimes, the MRS shift analysis was very close to positive. The MRS 0 to 1 dichotomy analysis is positive. So the point estimate is 1.64. So Filipino patients really did improve on treatment. So here's the prosaic analysis. It's probably because the prognostic factors in the trial. Overall, we know that there are 4 prognostic factors which turned out to be important. The 2 most important ones being age and your baseline NIH stroke score. And in the Philippines, we found that there was a much higher odds ratio of Filipino patients being more severe, being female, and having randomize later. So I think that this just shows you how if your treatment group was a higher risk of having a poor prognosis, had a higher dynamic range, it could show you a treatment effect. So is there life after chimes? Well, I've spent 6 years of my life doing chimes, and I think there's still a lot more we can learn. And one of the more important studies that we are taking up is this chimes extension study, which is run by my colleague, Dr. N.V. Ramani, where we're doing a blinded follow-up for 2 years after the index stroke. We're collecting data on the modified Rankin scale as well as recurrent events and death. My colleague Joey Navarro from the Philippines is also doing a chimes imaging study where we're looking at whether the initial CT scan can predict for outcome. What about publications? We've tried our best to go and sort of keep everybody informed of the results of chimes. I think the two most important recent papers have been the primary result and the recurrent vascular events result. We hope to analyze the database more and present more data in the future. Furthermore, we are undertaking other trials with Neuro8. We have started on a study of Neuro8 in vascular cognitive impairment, the Neurite trial. This is a 6-month study of Neuro8 in patients who have cognitive impairment after stroke. And we're going to recruit 100 patients from sites in the Philippines and Singapore over the next 2 years. Our colleague, Professor Valerie Fagan from New Zealand, has also undertaken a trial of Neuro8 in traumatic brain injury. Who are going to be treated for 6 months, and 140 patients will be recruited over the next 2 years. I know that there's a lot of interest into whether we can take forward the result of chimes to do another trial in non-acute stroke patients, perhaps a longer study with a 2-year extended follow-up period. It's a question of what is going to be the optimal primary outcome and whether we should measure recurrent vascular events as well. And many of our neurosurgical colleagues are keen on Neuro8 trials in hemorrhagic stroke. So to conclude, the meta-analysis shows that Neuro8 is probably useful in chronic stroke. Chimes, which was a well-designed and well-executed clinical trial to assess efficacy and safety in acute stroke, had a neutral outcome, but it did confirm the excellent safety profile of Neuro8. And there was, as you can see in the slides I showed earlier, further encouraging results on efficacy in terms of functional outcome and vascular events. And of course, as an academic neurologist, you know I'm going to say that we need more trials. But you are probably going to ask, what do we do right now? So that can be discussed later. So I finish off by thanking all my colleagues who took part in chimes. Professor Marie-Germaine Busser, who helped us lead the trial, Professor Jeffrey Donnan, who led the Data Safety Monitoring Board, and all my colleagues from across Southeast Asia, and the patients who contributed to this trial. So just to end, I think that this is only the beginning. There are many, many doors left to open for stroke recovery. And the other reason I show this slide is not because I look any better without a tie. It's because I wish to thank Moliac, as represented by Claire here, for working so well together with us in the academic field to go and sort of drive the development of traditional Chinese medicine in stroke recovery. Thank you very much for your attention.